filament with a live fertility assay, and it's in stat that shines half the stone and it starts to
It's a pleasure to introduce the next speaker, Janmey from the University of Pennsylvania, who will be speaking about the response of cells to nonlinear elastic substrates.
Sound okay? Okay, so thanks very much for this chance to show you some things that we're doing recently on looking at issues that are closely related to the previous two speakers, but done using somewhat different materials and different cell types. So the work I'm going to tell you is really done by these guys. Ilya Leventhal and Jeff Fifield have done a lot of the atomic force microscopy and mechanical measurements. I'll show you. She wanted to use microfluidics to make us some new materials with a gradient of stiffness that maximizes our ability to look at these mechanical responses. And Justin Weiner and Penelope Georges have done a lot of the cell biology to look at cell type specific responses to different ranges of stiffness. So what I want to tell you about are a few short stories of cellular response to the stiffness of the environment, and how general an effect this is, and whether or not this might actually be something that is biologically relevant in different contexts. So I want to show you a couple of different cell types to look at how specific these responses are. So I'll look a little bit at fibroblasts, which have been used a lot by in these kinds of studies, but also stem cells. It turns out we've already heard some mention of the effect of forces and mechanics on stem cell differentiation and proliferation rates, and these respond quite differently in their own way. And one of our interests has been looking at the effects of soft materials in the central nervous system, because this is one of the softest places in the body. And here there are some very so far a unique mechanical response of neurons that makes them different from all other cell types. So in principle, we'd like to understand how this happens. But secondly, we'd like to try to take some of the kind of lessons that we've learned from in vitro measurements and to try to design biomaterials that might actually be biocompatible and usable, and where you could exploit some of this mechanical information. So as should come to a surprise as a surprise to no one, but that's often neglected in cell biology, most tissues in our soft organs don't live on a flat, rigid substrate, and their lives are not dictated only by the chemicals that they ingest, or the genes that they have, but they're also typically embedded in three-dimensional meshworks of polymer matrices. Here are some fibroblasts crawling through a collagen matrix in a repairing tissue. And this nice colorized scanning electron micrograph shows you a blood clot, a clot of whole blood. These are the red cells inside a network of fibrin strands. This is a polymer that I'll mention again. This is the major polymer that assembles when your blood clots, and it assembles in this beautiful kind of modified polycondensation reaction that's initiated by enzymatic activation of bifunctional monomers. And the polymer chemistry and polymer physics of these things is pretty well studied. But this is a material that has tremendous potential for being used. And what's nice about this picture is that these little purple guys are platelets. And unlike the red cells, which are just passively trapped in this three-dimensional network that rapidly assembles in your blood to keep it from flowing out, these little platelets have done two things. They have activated in response to the same cues that made the fibrinogen polymerize, but they also have extended long arms, attached themselves to the filament, and then pulled in by an actomyosin contractile response. And in doing so, they make these little loci where several of these platelets have clumped together. But I hope it's visible that these relatively flexible, curly polymers have become taut in between places where these platelets have pulled on them. And the ability of cells to pull on the filaments to which they are attached has a very large effect on the stiffness of the material. So it's been known for a very long time that the stiffness of a blood clot that has platelets in it, or even just a pure fibrin network that has platelets in it, is about 10 times stiffer. Its Young's modulus is 10 times larger than if the platelets weren't there. Or even if the platelets were there, but you killed their motors using blebistatin or some other thing, the same kind of drug that Margaret showed us yesterday. And so the ability of these cells to stiffen the network is not because they're adding mass to the polymer matrix. It's simply because they're applying internal stress. And that internal stress ability to stiffen the network, it relies on the fact that these networks are intrinsically nonlinear elastic materials. They stiffen the more you strain them. And that's a feature that is not present in most synthetic elastomers, including the soft materials that most of us grow cells on. Okay, so those are the two lessons I want or the two kind of images I want to think about, and we'll look at these again later. Okay, so another motivation for looking at the mechanical effects is that it turns out that mostly bioengineers have measured the elastic moduli of any tissue that they can get their hands on. And there's a vast literature that we've made some weak attempts to kind of cull together. And in the literature, of course, confusing, but one of the things that is important, because as Jean-Francois mentioned a couple of days ago, all of these cells and even many of the tissues are viscoelastic liquids in some sense, or viscoplastic materials, that they act as very good elastic materials if you strain them on the time range of a few seconds or a hertz in an oscillatory measurement, and if you don't strain them too much. So in a roughly linear elastic regime of a couple % strain and about a 1 second time scale, there's a pretty large spread of elastic moduli of organs. This is organs from mammals, often from humans. And the softest of all materials, not surprisingly, is fat, mostly visceral fat, or the fat that's in your bone marrow where the stem cells live. So this has an elastic modulus of a few tens of Pascal, so only slightly stiffer than spit or mucus, but it still can sort of barely hang together. So you know what fat feels like. And as different tissues are compared, the second one of the softest tissues is the brain. The brain has an elastic modulus of a few hundred Pascal, and different tissues have these kinds of characteristic stiffnesses. And one thing that's pretty striking from the literature is that the variance in these kinds of measurements is surprisingly small. So the difference between organs from young and old animals, for example, or between chickens and frogs or something, those differences are bigger than the typical variance if you take a population of animals in the same state of health or the same age, for example. And when things go wrong, for example, when liver becomes fibrotic, the stiffness is sufficiently different, almost an order of magnitude, that it's palpable by your fingers. And of course, the classic example of this is the formation of malignancies in the breast. The breast is also a relatively soft tissue, which even in a pre-malignant form develops parts that are palpably stiff. That's why that's how self-exams are based on the ability to feel this difference with your fingers. And this difference relies on there being approximately an order of magnitude difference in stiffness. So it's generally been thought, I think, biologically, that these stiffness differences these stiffnesses are just an outcome. They're a consequence of the biochemical metabolic work that the tissue has to do. But it may also be, and it's beginning to appear to be, that stiffnesses or the elastic modulus of the tissue is a way for the cells in there to decide how to behave. And when the tissue stiffness goes wrong, as it does in cancers or fibrotic disease or scar formation, the cells in that tissue, even if they are genetically normal, start to behave abnormally. Okay, so that's the idea to have in mind. We've seen several examples of this, and this field was largely kind of brought back to life by a paper by Pelham and Wang about 10 years ago. This is our version of their experiment, where they looked at simply the actin cytoskeleton, or the morphology of the cell. This is a garden variety fibroblast grown on matrices very similar to what Cindy King showed us a little bit ago, that this increase in elastic modulus causes these types of cells to spread more and to start assembling stress fibers. So we've looked at other aspects in other cell types, and here's one example of what we've started to look at lately. This is work by a new postdoc like Shang UT, who has learned to do total internal reflection fluorescence from the interface between a soft material and the aqueous substrate. In this case, it's a very lightly cross-linked PDMS substrate. And what we're looking at are the focal adhesions, the same things that Margaret showed us about, but as a function of how stiff they are. So here's a GFP vinculin molecule that's expressed by these fibroblasts, and they're either put on very stiff or very soft materials, and no forces are being applied to them. The cell is just doing whatever it wants to chemically identical surfaces. The only difference is this is stiff, this is soft, and here we're only showing a quarter of the cell because it's so big, because it spreads so much. And these focal adhesions are very large and elongated, as Margaret showed us, but when you put them on soft things, they're neither elongated, maybe a couple of them are. They're smaller and they're round, but there's more of them. And they're very dynamic. These dance around even in real time, whereas these things are relatively stable. So what determines that switch is still very much unknown. In order to study this, we've recently started to try to improve the ability of doing these experiments, because it's a bit of a pain to make, as everyone who's done these knows, to make a bunch of these gels and to look at things separately. But it turns out you can use microfluidics to make gels with a gradient of stiffness by changing the concentration of the cross-linker for the linear polymer from which the gel is made. In this case, it's one of these polyacrylamide gels, but in principle, this can be any material that you can push through a microfluidics device. So in our case, we make little gels that are about 1.5 millimeters in width, 100 microns thick, and a couple centimeters long. And you can put whatever adhesive protein you choose to put on there by the standard chemistry. And this is actually one of our early attempts, and it's not a particularly steep gradient. It goes from a few tens of kilopascal, or a few kilopascal, to about 50 kilopascal over this 1.5 millimeters. So that's a steep enough gradient so a big cell will feel something that's about 4 or 5 times stiffer on its front compared to its back. But we have this whole range of stiffness where we can put a dish of cells in the same chemical medium, the same everything, and look at how stiffness affects things. And here's a typical example. Here's one of these gradients. In this case, these are melanoma cells that have this similar nice reaction. They're small and round and kind of undeveloped on the soft part. They become extended and make stress fibers on the hard part. And if you knock out a particular actin cross-linking protein, the cells lose the ability to do that. That's something I wish I had time to talk about more, but many people now are beginning to look at which proteins are important for a cell to do this trick. And it turns out that there are very specific pathways you can stop, and the cell will no longer respond to stiffness cues. Okay, it'll still be alive, it'll still be okay, it just won't respond. So
which actin crossing protein is it?
That's filament A.
Oh, it is.
And it turns out that filament A blocks that activity only when the cell is binding through collagen receptors. If you give it fibronectin to spread on, it doesn't need the filament. Okay, so one of the other things that's come as a bit of a surprise is we've started to do, how do I get this to work? Atomic force microscopy with this nice little needle by making a gel, poking on the cell that's sitting on that gel, and then poking on the gel right next to it to get an idea of the relationship between the cell's own stiffness and the stiffness of the material around it. And these measurements, there are technical issues of doing them that there's no sense in going into, but the bottom line is that we can use relatively simple models and get a surprisingly accurate reading of the Young's modulus of these materials, and again about a 1 hertz frequency range and on the order of a 10% strain. Okay, so that's that. And the result is this, that here's something that people have seen since they started first to do atomic force microscope measurements of cell stiffnesses. If you take a very rigid material, two things are pretty evident from, in this case, this is a fibroblast protruding on a plastic surface. One is that on this kind of pixelated stiffness map, where each pixel is about 50 by 50 nanometers, there's a fairly large heterogeneity of stiffnesses. So some parts of the cell look like there are a few tens of kilopascals. Some parts, more in the interior of this protrusion, are about 4 kilopascals. It's relatively heterogeneous, and the upper limit seems to be someplace around 20 kilopascal, for example, for this cell type, sitting on something that's infinitely stiff, that's gigapascal. And this is just the tip of the cell. The nucleus is way back somewhere else. Okay. But if we do the same thing and we put it not on an infinitely rigid surface that the cell would never see in vivo, but on a matrix that's relatively appropriate for the tissue for where these things come from, two things happen. One is, here's again a color map of the stiffness. Here's the outline of the cell, and this is the gel around it. One is this, a lot of this heterogeneity goes away. The cell is relatively uniformly stiff in one of these protrusive areas. The protrusions are also much more blunted. But the cell is approximately as stiff as the gel outside it. And that's not a coincidence, at least for this cell type. If we change the substrate stiffness and look at how the cell's own stiffness depends on the substrate stiffness, for fibroblasts, mesenchymal stem cells, astrocytes, many other cell types, but not all cell types, the stiffness scales with the cell stiffness scales with the gel stiffness up until a point where the cell can't get any stiffer. You can make this substrate as stiff as you want, and eventually the cell will peter out. But during a very large range of substrate stiffness, which is approximately the range in which organs actually are, the cell is tuning its own stiffness to match its substrate. And what's curious about this is that it's not proportional to the substrate stiffness. It's equal to the substrate stiffness, at least for these cell types. That's not always true. Some cell types maintain a constant stiffness, couldn't care less about the gel stiffness. But many tissue-forming cells do this. Okay. So as I mentioned, not everything works the same, and one of the most interesting deviants or rebels from this kind of behavior is the neuron. Motor neurons and cortical brain neurons have exactly the opposite reaction to substrate stiffness. They will extend their long so here's a process that might eventually become an axon that typically grows relatively rapidly and branches. But here's the cell, here's one of these neurons growing on a soft material, 100 Pascal or so, so in the range of a normal brain, that does this very nicely. It extends a lot of these red stained microtubule pores, or the core of the axon, but off of it emanate these large branches of green labeled actin, one of which will become the growth cone that will drive the thing forward, and some of which will become branches to allow these neurites to explore more of their space. As the substrate stiffness gets stiffer, but no chemical change happens, as branching density goes down, the rate at which these extensions go out also slows down. And even on very rigid substrates, you can get them to grow, but they'll very rarely branch, and they'll grow somewhat reluctantly. So this is the opposite trend. As far as I know, this is the only cell type that grows out faster on soft materials than hard materials. Its neighbors in the central nervous system, astrocytes, act just like fibroblasts. They're round and unstructured, but metabolically perfectly happy. So this is a surprising thing. These cell types, which ordinarily have beautiful stress fibers, have very well assembled intermediate filament systems, when you put them on something soft, they have zero stress fibers that we can see, and they depolymerize their intermediate filaments, but they're metabolically still happy. So one of the things we did a few years ago was to take this quantitative information, look at the range of stiffness where things like astrocytes get activated, which is something you don't want to do in a central nervous system injury site, and see if we can find materials that are soft enough so they're promoting of neuron growth, but not very excitatory for glial cells like astrocytes. And in fact, you can find materials like this. So here's an example of taking a whole explant of the brain cortex of a mouse. So all the cell types in the brain, and plop that down onto a culture dish that has either a very soft gel or, as I'll show you later, a stiff gel on it, and then go away for 3 or 4 days and come back. And then stain the cells that are growing on there, either with a red stain for neurons, or I'll show you later, a green stain for astrocytes, for other glial cells. So in this case, here's a very soft gel that was treated with all the cells in this tissue, and mostly all that we see growing on top of these materials are neurons, red-stained neurons. If we take the same preparation, just put it on a stiffer gel, we see mostly green-stained astrocytes. There are still some neurons growing, which is good, but they're growing on top of the astrocytes, which are soft compared to the material underneath them. So we got kind of enthusiastic about this because it does suggest that by tuning the stiffness of the material, you can select which cell type will prevail, or you can at least find a mechanical environment that will be promoting for neurite outgrowth. And it does work in 3 dimensions in a material that you could imagine you could implant into the brain, one of these fibrin gels. So fibrin, the same material, blood clotting material I showed you before, if you purify it, you can make gels that are either stiffer than the brain, in which case only or mostly these green labeled astrocytes grow. But if you make the gel softer and softer, the red labeled neurons start taking over, and they make these beautiful, long, extended collections after about 6 days that require the soft matrix to grow. So these cell types respond to 3-dimensional fibrin gels mechanically, very similar to what we would predict by looking at their behavior on these soft polyacrylamide gels. So I want to take one little brief detour to show you a movie, because everybody else has such beautiful movies. And to show you one of the other things that is wildly dependent on substrate stiffness. So this is cell division, cytokinesis in this case, of 3T3 fibroblasts. So here's again one of these gradient gels. We're just looking at time lapse of cells dividing on either a stiff part of the gel, in this case it's 4,000 Pascal, or a very soft part of the gel, 300 Pascal. So on the stiff part, the fibroblasts move around and they divide pretty much the way that they would on glass. I think if you showed this to somebody who worked on fibroblasts, there's nothing surprising here. This is what they look like on glass. But this is a gel that's about 4,000 Pascal. So meanwhile, and this movie takes about a day and a half or so, or maybe somewhat less. Meanwhile, their neighbors on the other side of the gel are doing something completely different. Watch this guy. They round up and they explode. And they are not able to put these traction forces to get themselves to spread out in the spindle shape. They just develop internal tensions. They're still able to do mitosis, but their cytokinesis program is all messed up by not being able to distribute the forces. I think these... Sorry?
Can you play the movie again?
Yeah, I think these cells are... no, I can't, Margaret. Oh, yeah, here we go. Okay. So watch this guy.
Oh, my goodness.
And one of the curious things is they never divide into 2 pieces. They always divide into at least 4, 2 of which I think have the nuclei that will become daughter cells, and the other 2 or 3 or 4 are cytoplasts, which are just pieces of of membrane-bounded cytoplasm, which are still alive and kicking, and they'll eventually fuse with another cell body somewhere. So that's where we are on that. But that cell division in this case is specific for a laboratory strain cultured cell that divides perfectly fine on soft materials. Many cell types, in this case, these are some primary cells taken from either newborn or embryonic mice. These things have a very different response. So in this case, several cell types, including mesenchymal stem cells, stop dividing, stop proliferating when you decrease the stiffness. So here we're going from stiff to soft on this scale. So as the shear modulus decreases to about 1 kilopascal, several cell types, but usually they have to be primary cells. This does not work with immortalized cell lines. They proliferate very well if you put them on something stiff, even when they're subconfluent. They can stay subconfluent, but as you decrease the stiffness, they stop dividing, and they stop expressing cyclin D, and many of the markers that are specific for cell cycle checkpoints don't get satisfied. Many of them are, some of them are not. But anyway, the cell stops dividing. But it's still okay. It's still perfectly happy. I'm going to skip over this just to show you that this same thing I showed you for these embryonic fibroblasts also happens for mesenchymal stem cells, the stem cells that come from your bone marrow that can eventually differentiate into many different cell types, which Disher and other people have shown are also very strongly sensitive to stiffness. Okay, but these cells also, if you put them on something soft, they will sit there for weeks, months, and not divide, and they're still happy. And one of the reasons we can show that they are still happy is by making one of these little sandwiches. So the experiment is like this. We've grown the cells on a soft shell for several days, and they've become quiescent. So here are two different cell populations on soft materials. So on day 3, we put on top of them a little hat. And the hat has either another soft shell that's just as soft as what they're sitting on, or a stiff shell, or a piece of glass, something that's really rigid, where normally they would be divided. As soon as we put the little top on them, if it's a soft shell, the cells change their morphology a little bit. This is exactly the same field, and these are the same cells. So they have squished a little bit, because now we've made them have adhesion sites both on top and bottom, so their morphology has changed. But they didn't divide, and they're not going to divide. But as soon as we put something rigid on them, there's no chemical stimulus. They just have a rigid surface on their heads now. They immediately start to proliferate in an almost synchronized way. So all they're waiting for is a mechanical signal in order to divide. So what that mechanical signal is and how it's sensed is something that we'd like to figure out. Okay, so this leads us to the nonlinear part. So how does any of this stiffness sensing that we've looked at, or this mechanosensing, work? The kind of zero order cartoon picture is that at the membrane, there have to be, presumably the sensing happens at the membrane, because that's its interface with the outside world. So transmembrane proteins are bound to extracellular proteins, and on the inside of the bilayer, they are attached also to large adapter molecules of these focal adhesions, of which there are many. And these things are linked to the three-dimensional cytoskeleton, which is eventually linked to the nucleus. So if you put a force on there, or if either you pull from the outside or the cell contracts from the inside, a tension is now transmitted through this series of springs. And it's tempting to imagine that the way the cell knows whether the outside is stiffer than the inside is it asks which spring is the softest, and therefore which spring is the one that's most stretched. If the cell is rigid and the outside world is soft, it's the outside world that's going to get deformed and potentially activate some signal. If the outside world's rigid, more rigid than the inside, the cell will yank on this series of springs, and the one that will open up is one inside, and that will send a different signal. The signals that the cells use to respond in order to do these morphological changes are probably exactly the same signal transduction pathways that they use when you give them a growth factor or some other chemical stimulus. It just got initiated by the force, I think. It's also, of course, possible that these tensions are transmitted through the bilayer to, for example, stretch-activated ion channels or other things that can activate these pathways as well. But so we've started to try to look at what are the mechanical properties of these sort of imaginary springs. And one of the things that's now fairly commonly seen is that if you look at the most simple nonlinear behavior of materials that are characteristic of the stuff inside the cytoskeleton and out in the extracellular matrix. So collagen, cross-linked actin, fibrin, which I just showed you, intermediate filaments like vimentin or neurofilaments. So both the extracellular polymers, collagen and fibrin, the intracellular polymers, intermediate filaments and actin, they all have this characteristic that their shear modulus is a very strong function of the strain. They are very soft and compliant at low strains, and what is low for these things depends on their own stiffness and their cross-link density. But low strain typically is a few percent or maybe 10%. But as you increase the strain, the materials stiffen, and they stiffen a lot. They stiffen orders of magnitude. And this is a reversible process. If you now take something that's been stretched and is now stiff and relax it, it's soft again. This is something that doesn't happen in polyacrylamide or other elastomers. And this is a feature that cells and tissues, I think, exploit because it means that if you want to locally stiffen your cytoskeleton, rather than having to polymerize more actin or put in more crosslinks, all you have to do is to activate some motors and put an internal tension on that. Putting an internal tension on the material rides you up this strain stiffening curve in a reversible way. If you relax the motors, it gets soft again. And I think this is the explanation for why platelets stiffen blood clots. Blood clots formed at rest are relatively soft and compliant. If you put in internal contraction with platelets, that's the same as taking a rheometer and putting an external shear strain on it. They pull the slack out of the filaments, and the material gets stiffer again to its next attempt to be deformed. So where this nonlinearity comes from has luckily for us experimentalists attracted the attention of theory people, and there are two really interesting models that can account for many of these aspects. One is that many of these polymers are very different from synthetic elastomers in that their persistence lengths are very large. The persistence lengths are on the order of the mesh size of the network, so hundreds of nanometers or a micron or so. And that means that the intrinsic force extension curve of the elastic elements inside that network, those force extension curves are intrinsically nonlinear. So by putting a shear, any kind of deformation on these networks, it takes much more force to stretch a filament than it does to compress it, and that gives rise to nonlinearity in the elastic modulus. And there's a second model, proposed, I think, first by Patrick Long, but looked at by several groups, which says no, it's not exactly that the material properties of the polymer have to be nonlinear. It's simply the fact that you have materials that are made from stiff. The movie won't play. That are stiff fibers. As you take a material that's made out of fibers like this and deform it, there is a balance of restoring forces that comes from filament bending and filament stretching. And because bending is soft and stretching is hard, the more you align the filaments in the direction of the deformation, the more you transfer the forces onto stretching modes, which are stiffer. And that accounts for this increase in stiffness of biomaterials made from stiff polymers. So which of these models actually accounts for our materials is still up for some investigation. So now back to the cell business. The reason this is, I think, interesting for mechanical sensing is because different cells pull with different amounts of enthusiasm. So the endothelial cells we looked at appear to pull with very large forces, hundreds of kilopascal. But there are other cells, like neurons, that pull very little, a few tens of pascal maybe. So some things pull a little bit, and therefore we put small strains on a material. Some things pull a lot, therefore put big strains on the material. So if you have something that pulls a little, and you put it on two materials, for example, a strain stiffening fibrin gel and a linear polyacrylamide gel, if you formulate those gels so they have the same linear elastic modulus, a weak cell like a neuron will see them both as soft. But a hard pulling cell that puts a large deformation on these materials will still see the acrylamide gel as soft because it's linear. Its elastic modulus is the same all the time. But if it pulls far enough to engage the strain stiffening regime in the nonlinear gels, it'll see these as stiff. That's at least the hypothesis. And this is both, I think, borne out by experiments, and it's also a bit of a problem for our attempts to use them biologically. So here again is this experiment where we've now looked at the projected cell area, similar to what we've seen for endothelial cells and other cell types, but in this case it's for mesenchymal stem cells. These also have this beautiful stiffness response. They get bigger as you increase the elastic modulus, but these acrylamide gels are never going to be useful biologically. So we thought, okay, great. We'll look at what happens if we put them on fibrin gels, very soft fibrin gels. Can we do the same thing we did with neurons? Can we make a fibrin gel where we could store stem cells for a month and everything would be cool? And unhappily, it doesn't work. If you make a fibrin gel that has a linear elastic modulus of a couple hundred Pascal and put stem cells on them, they'll elongate, and they are just as, they see these materials just as though they were 100 times stiffer. Why is that? It's possible that it's a chemical thing. So we've looked at taking polyacrylamide gels and giving them as the cell attachment site the polymers that make the fibrin network, right? But not a polymeric network of fibrin with its own intrinsic nonlinearity, but just the building blocks of the fibrin network. Either the enzyme-activated monomers, or the thin protofibrils that will eventually make the network, or the big fat fibers. As long as as you trap the reaction before the gel point, you can make soluble filaments and covalently link them to the top of the surface of the polyacrylamide gel. Now the chemical information comes from the fibrin, but the mechanical information comes from this linear elastic polyacrylamide gel. So if we put the chemical ligand on an acrylamide gel, if we put either monomers or protofilaments on it, the cells still see it as soft. If we put a three-dimensional network on top of the polyacrylamide gel, they see it as hard. So they're somehow engaging this three-dimensionality. And this has also some consequences in the kind of long-range mechanical sensing that cells might do if you put them on a nonlinear elastic material, which is its normal environment in the body, compared to an elastic material. So we've done very similar things to what we've already seen from traction mapping experiments for polyacrylamide, but we've done a very kind of coarse-grained deformation map in these fiber networks. So here's an electron micrograph of the gels on top of which, or inside of which, these cells are put. So this length scale is about 2 microns. This is a typical meshwork of a fibrin gel before the cells do their thing to it. And in order to look at the deformation, we've covalently modified a few of the monomer units that go into these filaments with biotin, to which we can attach streptavidin, which is just a ligand for biotin-coated fluorescent beads. So we can attach, you probably can't see this, there's a tiny little red dot here, which is the size of a bead. That's about the same size as the diameter of the fiber, and we put a couple of these beads in these networks covalently, and then we watch the beads move just as we've seen before. And the reason it's sort of coarse-grained is this mesh size is pretty big, and also the movement. It turns out that the movement of these beads is much larger and carries out much farther than ever happens in these polyacrylamide gels. But I'll skip over this because you've already seen how this method works, and just show you a typical example. So here is a cell body, and this cell body is about 100 microns long. This is a stem cell. Here's a little bit of a blow-up of it. And here's one of these deformation maps. We do the deformation maps just the same as Margaret showed us yesterday. We put some cells on these gels. We allow them to contract and then we poison their contractile machinery with blebistatin or depolymerize their actin, and then the cells will relax. They'll get rid of their tension, and by looking at how those beads move, we can figure out how much the cell deformed it. Okay, so a lot of these beads have undergone movements, but they undergo movements in a kind of an asymmetric way. So this plot is the farther we are from the origin, the larger this displacement is. So the beads will move about 6 microns at the edge of the cell, and there's an asymmetry. If here's the long axis of the cell, beads are moved, and therefore the deformations are larger along the long axis of the cell than along the short axis, but they're really contracting everywhere. And they contract the matrix surprisingly far away. So here is one example of a kind of an average bead displacement measured circumferentially everywhere in a little zone that is either within 250 microns of the edge of the cell or more than 250 microns away. And even up to 250 microns away, beads are being moved a couple of microns. And we can tell that because this is this typical thing. If we poison the contraction with blebistatin or cytoplasm, we get these motions. But even 400, 500 microns away, the beads are still noticeably moving. It's only when we get past a half a millimeter that we can't see statistically significant movements. And here's kind of an interesting thing. If you don't depolymerize the actin, but instead you depolymerize the microtubules with the cortisol, the cells pull harder. This is a well-known phenomenon that depolymerizing microtubules often releases small GQPases that will activate myosin even more. And by doing this, by putting active contractions, now we can actually see statistically significant deformations half a millimeter away from the edge of the cell. So how is the cell able to do that?
So wait, your units there were millimeters. You didn't mean
on
the x-axis.
Oh, yeah. It's the
Microns.
Alt key. Yeah, I would no.
Okay.
Millimeters would be pretty amazing. No, microns. Sorry, these are microns. And may be relevant to whether cells are controlling stress or strain. The softer we make the matrix, that is, the less we the lower the fibrinogen concentration, or let's look at it the other way. The larger the fibrinogen concentration and the bigger the stiffness, the smaller the magnitude of the displacement is. But again, we can see relatively good displacements 400, 500 microns away from the cell, especially when the gels are soft. So why is that interesting? It's interesting, I think, for 2 reasons. When the cell is able to contract the gel, it's also able to ride up that stiffness sense, that strain stiffening curve. So here's one attempt to do this. Here's again an atomic force microscope stiffness map of, here's the cell outline, but what we're looking at is the stiffness of the gel around it. When we began, if there wasn't a cell, the AFM would give us a very uniform stiffness of the fibrin gel that's about the same stiffness that we would measure in a rheometer. In this case, I guess about a little less than 1,000 Pascal, a couple hundred Pascal. But if we look at the local stiffness near the cell, the stiffness near the cell is significantly bigger than the stiffness far away. And again, if we poison the contractile machinery, areas near the cell become soft again. They regain the elastic modulus of the bulk gel around it. So the cell, by putting in contractions, is not only transmitting tension along its matrix, it's also locally stiffening the matrix. So cell anywhere in the zone that a neighboring cell has been pulling feels a stiffer substrate than it does far away. So this has at least one consequence that's related to cells signaling to each other, or cells mechanically sensing each other, in that as we make, it's something we don't see very well in linear polyacrylamide gels of the same elastic modulus, we see much better alignment of the long axes of cells when they're on these strain stiffening fibrin gels with these very long range strain fields. Here's a bit of as an example of how far away cells can sense each other's presence. So these are still very sparse populations of cells. They've never come together and touched each other. They're not laying down the chemical path that we know about. They're just feeling each other mechanically. And you can tell that they felt each other mechanically because if you look at the spread area of the cell that is within 200 microns of its nearest neighbor, those cells are bigger than cells that are 400, 500 microns away. So they don't have to touch a cell. They can just be near a cell and become bigger, presumably because they felt some stiffness. And they become more oriented. So the angle between the long axes of the cell is quite small if two cells are within a couple hundred microns. It dissipates to sort of a random distribution only when you get more than a half a millimeter away. Okay, so let me finish there by concluding that in a very cell type specific way, cells respond to mechanical properties of their substrate, but they respond differently, presumably because they have different levels of force that they apply to materials, but also they have different signaling pathways. So it's very difficult to predict what one cell will do and another cell might not. But the contractile cells, the ones that are the most efficient at yanking on the materials, one of the things that they do to the natural materials is locally stiffen it. And that local stiffening, as well as the tension that they apply, is one of the stimuli to which their neighbors respond, and that this mechanical thing is a way for cells to respond to cues that are hundreds of microns away, much farther away than could be accomplished by touching. Thanks for your attention.
Thank you very much. We have time for one or two quick questions.
So in understanding how strain fields propagate so far in except the Fibrin network as you've prepared it, I mean, can we think of it as maybe that individual long Fibrin fibers have a very high stretching modulus or bending modulus, however the network itself it has a low modulus, so basically I can take a rope and pull it through the network and a long way away it's going to follow. That's easy. However, the fiber itself is very rigid.
Yeah, it's possible. So we've started to try to visualize individual fibers. There are, as far as I know, no fibers that are 400 microns long. But the strains are not big enough that we have oriented the fiber enough to become, the material, for example, has not become locally birefringent at places where there is cell-cell contact. So I can't rule out the possibility that two cells are attached to each other by a rope that they're pulling on.
Yeah.
But it doesn't seem, we have no evidence for that yet. It seems to be more a collective phenomenon.
I have a question on stiffness. So do you think that does he prefer the stiffness of the matrix which is comparable to cell's own stiffness? Like, is there any correlation between this thing has been observed?
Yeah. So far, in the limited number of cases, that the range of stiffness that cells respond to is similar to the stiffness of the organ where that cell came from. So, for example, materials that a fibroblast would find completely soft because it comes from a stiffer place, a brain cell would find as unacceptably hard. There is some. It's tempting to think that that's true. That at least for primary cells that have a memory of where they came from, something is optimized from at the stiffness of the material where they came from when it was healthy.
Okay, I have one more question on the simple mechanical model, spring model that you have shown.
Yeah.
So it's the model will predict the cell sensing depends on the deformation, as you said, that how much pulling the spring inside the cell and the spring outside the cell, because the force would be same, force would be balanced, and that would be the same. So do you think that deformation, cell mechanosensing is deformation related? What's the common belief?
Yeah, I have no idea. Yeah, I was going to ask you that. I don't know. Nothing in our experiments gives us a clear example. Some aspects look like they're strain dependent. Some things look like they're stress dependent. So I haven't seen a general rule.
So you said that
neurons like to grow on soft things better, and you said something about the actin filaments coming out of the microtubule core. But can you say something about why the neurons like the softer?
The only thing, well, why they like it, I mean, what it is about their signaling thing that makes them respond. Well, two things. One is that one might think that a neuron wants to grow in a place that's soft because that's how they're guided during brain development. They want to go along, for example, a radial glial cell, which is a soft guide to know where to go. There's no reason for a neuron to go into someplace stiff because that's not going to be any place that it needs to be. From a signaling point of view, it turns out that neurons have a response to activated Rho and focal adhesion kinase that's the opposite to the response that other cell types have. So on the level of signaling, there's a chemical pathway that neurons seem to use differently from other cell types.
So wouldn't you be able to measure or determine whether it's strain or stress stiffening by measuring the modulus of the cell on those fibrin matrices with different linear moduli, comparing the stiffness of the cell and the local strain field? So if it's stress dependent, the cell stiffness should be the same, and if it's sensing strain, the contraction of the fibrin gel should be constant. So you showed that nice measurement of cell area that was constant, and so you want to know if the modulus of the cell in those different conditions is the same or different.
That's a good idea. Okay, yeah, great.
Thank you very much. The next speaker is Lisa Manning from Princeton University. There's a change in title to her talk.
Change the dip?
Yeah, change the dip now. Okay, so you don't have
How do I get this out?
it did it. It pops up by itself.
Again?
Yeah, so like so.